Bismillah ar-Rahman ar-Rahim. I would like to welcome you all to this simplified lecture on coronavirus COVID-19 variants, and in today's lecture I am going to concentrate on the UK, South African and the Brazilian variants of coronavirus COVID-19. This diagram, which I have drawn or constructed, will simplify the different variants of coronavirus COVID-19, and we are going to start with the UK variant. However, before talking about the UK variant, I would like to refresh our memory regarding the attachment and entry of coronavirus COVID-19 into the human cell. So what you see here, which I'm pointing in this diagram, which I'm pointing at with my laser pointer, you can see the coronavirus COVID-19. So you can see the viral envelope in blue color, coronavirus COVID-19 RNA in black color, which I'm pointing at with my laser pointer. And then, as I mentioned previously, the coronavirus COVID-19, it has spikes or S protein, which is this one. You see it here. I'm pointing at it with my laser pointer, and you can see it in red color. As we have stated previously, that the coronavirus COVID-19 attached to what we call it a cell receptor, named as ACE2 or angiostatin converting enzyme 2. So you can see the angiostatin converting enzyme 2 in green color. So this is the human cell, this is the cell membrane in red color, which I'm pointing at with my laser pointer, and you can see the ACE2 receptor or the angiostatin converting enzyme 2 receptor in green color. And then you can see the attachment of the coronavirus COVID-19 spike, which is in red color, is attached to the ACE2 receptor, which you see it here in green color. So the replication of coronavirus COVID-19, it starts with the attachment of the spikes or the S protein of the coronavirus COVID-19 to the ACE2 receptor on the cell membrane. Also, you can see here this triangle in blue color. This is, we call it transmembrane protease. You see it here, this triangle or blue triangle. This is, we call it transmembrane protease. This enzyme is produced by the cell. The function of this enzyme, which is produced by the cell, it's a protease enzyme. It will help in the cleavage of particular protein or peptide of the coronavirus COVID-19, and it will help in the entry of the coronavirus COVID-19 into the human cell. So you see it here. This is the TMPRSS2 or transmembrane protease. You see it here as a blue triangular. This enzyme helped in the cleavage of the spike protein and proteins of the coronavirus COVID-19, and this will facilitate the entry of coronavirus COVID-19 into the human cell. Then once the virus enter the human cell, then, as described previously in much more detail in previous lecture, after the entry of coronavirus COVID-19 into the cell, it will followed by the replication. Then we have the assembly, and then the release of the mature coronavirus COVID-19 from the cell. You can see here a mature coronavirus COVID-19 released from the human cell. I have described every single step of the replication of coronavirus COVID-19 in a previous lecture, which you can refer to it. Okay, now after refreshing our memory regarding the attachment and the entry of coronavirus COVID-19 into the human cell, and the attachment of the coronavirus COVID-19 utilizing its spikes into the ACE2 receptor, or the angiostatin converting enzyme 2 receptor. Now we are going to start to talk about the new variant of the coronavirus COVID-19. The first variant which has been emerged, we call it UK variants, or P117 variant of coronavirus COVID-19. The UK variant of coronavirus COVID-19, it has 3 different mutations. So the UK variant of coronavirus COVID-19 B117, it has 3 different mutations. The first mutation of the coronavirus UK variant, we call it mutation N501Y. Now, this mutation in N501Y results in amino acid substitution in amino acid number 501. So, what will happen as a result of this mutation, which we call it mutation N501Y, which has been seen in the UK variant of coronavirus COVID-19? What you see here, what I was describing previously, you see here, if we enlarge this picture, if we enlarge here, you see it here, we have the spike, which is in red color. This is the spike in red color, okay, attached to the ACE2 receptor, which is in green color. And as you know that the ACE2 receptor is located on the cell membrane. So what you see here is, we have, this is the spike protein, which is in red color. I'm going to get it with my laser pointer. This is the spike protein in red color, which I'm pointing at with my laser pointer. The spike protein of coronavirus COVID-19 consists from 3 different components. The first component, we call it S1, which is this one. I'm pointing at it with my laser pointer. The second component of the spike protein of coronavirus COVID-19 is, we call it S2, which I'm pointing at with my laser pointer. The third component of the spike protein of coronavirus COVID-19 is what we call it the receptor binding domain, or RBD, which you can see it here in blue color. You see it here, receptor binding domain, RBD. You see the RBD in blue color, okay? And then this spike protein, which consists, as I mentioned, from S1, S2, and the receptor binding domain, or RBD, will bind to the ACE2 receptor, which you see it here in green color. So this is the ACE2 receptor in green color, and this is the cell membrane. So what you see here is the enlargement of this area. So okay, if you can draw it right here, a circle, you see this area. If you enlarge it, you will see this diagram. So we have the spike protein of coronavirus COVID-19 consists from 3 different components, S1, S2, okay, and you see the first, and we have the receptor binding domain, which you see it in blue color. Abbreviation of receptor binding domain is RBD. And then this spike protein will be attaching to the ACE2 receptor, which you see it here in a green color, which I'm pointing at it with my laser pointer. And here we have the cell membrane. Okay. Now, this is what happens in the original coronavirus COVID-19. However, as a result of a mutation N501Y, as I mentioned, this mutation in N501Y result in amino acid substitution in amino acid number 501. I'm pointing at it with my laser pointer. As a result of this mutation, as a result of this N501Y mutation, this mutation results in the change in the shape of the receptor binding domain. You see it here, the blue color. So here we have the S1. This is the spike protein. Okay, we have the S1 as I mentioned previously. We have the S2, which I'm going to go with my laser pointer, and then we have the blue color, which is the receptor binding domain, or the RBD. As a result of the N501Y mutation, this will result in change in the receptor binding domain. So you can see here that there is a for example, like change in the shape, or that there is a thickness. You see it here in the receptor binding domain. So you see it here. For example, it's becoming thicker. This change in the receptor binding domain of the spike protein of coronavirus COVID-19 will result in more binding and interaction of the spike protein to the ACE2 receptor, or angiostatin converting enzyme 2 receptor. Again, in the UK variant, we have three different mutations. The first mutation, which we call it N501Y, this mutation, or nucleic acid mutation, result in amino acid substitution, and to be more precise, in amino acid number 501 of the spike protein of coronavirus COVID-19. This mutation, or N501Y mutation, will result in change in the receptor binding domain, or the RBD, of the coronavirus COVID-19, which you see here in blue color. And you can see that this change in the receptor binding domain, which I'm drawing now a black circle around it, for example, it will become thicker. It means this change or mutation or change in the receptor binding domain will result in more interaction between the spike protein and the ACE2 receptor. You see here, more tight binding or more interaction between the RBD and the ACE2 receptor. So more binding between the RBD and the ACE2, or angiostatin converting enzyme 2 receptor. Now, because that there is more binding and interaction between the RBD of the spike protein and the ACE2 receptor, then more virus, or more coronavirus COVID-19, will enter into the human cell. So we have increase of the virus entry. Now, because there is more virus enter the cell, so what that mean? Because more virus enter the cell, this will result in the increase in the replication of the coronavirus COVID-19. Increase in the replication of coronavirus COVID-19 inside the human cell, it means that it will increase the coronavirus COVID-19 viral load in the lung, or in the pneumocytes cells of the lung. Increase of the viral load of coronavirus COVID-19 in the lung, it means what? It means that more virus will be released during the sneezing process. So more virus will be released during the sneezing, so more virus in the droplet, so there is more transmission of the coronavirus to the other healthy individual. So you can see that because that there is a mutation, which we call it a mutation N501Y, in the UK variant of coronavirus COVID-19, or P117, this mutation results in change in the receptor binding domain, which you see it here, change in the RBD, or receptor binding domain, of the spike protein of coronavirus COVID-19. This change in the RBD results in more interaction of the spike protein, to be more precise, more interaction of the RBD or the receptor binding domain to the ACE2 receptor, which you see, this is the ACE2 receptor, which you see here in green color. This intimate binding or interaction between the RBD or receptor binding domain of coronavirus COVID-19 to the ACE2 receptor will result in more virus entering the cell, so increase the virus entry or increase the entry of coronavirus COVID-19 into the cell. This means increase in the replication of coronavirus COVID-19. Increase the replication of coronavirus COVID-19 inside the cell means the increase in the viral load, especially for example in the lung. Then increase in the viral load, it means that more virus will be increased during the sneezing process. It means that more virus will be in the droplets, so it means that the transmission of the coronavirus will be increased. So it's a very interesting. So this is the first mutation detected in the UK variant of the coronavirus COVID-19, which we called it N501Y mutation. The second mutation of the UK variant of coronavirus COVID-19, we call it mutation Hv69/70. This mutation, as a matter of fact, it was a deletion in the gene that codes for the spike protein of coronavirus COVID-19. This deletion in the gene that codes for the spike protein of coronavirus COVID-19 resulted in amino acid substitutions of the spike protein of the coronavirus COVID-19. This amino acid substitution occurred in amino acid number 69 and 70. Let's go back to our famous diagram. You remember what we have said previously? That the spike protein of coronavirus COVID-19 consists from, remember, we have S1, which is this one. So the first component of the spike protein of coronavirus, we have S1. Then we have the S2 component of the spike protein of coronavirus COVID-19. And then the third one, which you see it here in blue, is the receptor binding domain of the coronavirus COVID-19. Okay, so what changes resulted from this or second mutation in the coronavirus COVID-19, and to be more precise, in the UK variant, or the second mutation in the UK variant of coronavirus COVID-19. The second mutation, or the HV69/70 of the UK variant of coronavirus COVID-19, resulted in structural changes of the S1 and the S2 part of the spike protein of the coronavirus COVID-19. You see it here? You see it here in the yellow color? You can see that there is structural in the morphology, that there is changes in the structure of the morphology of the spike protein of the coronavirus COVID-19. You see it here in the yellow color. So the deletion or the mutation, which we call it HV69/70, in the nucleic acid or the gene that codes for the spike protein, which resulted in amino acid substitution, and to be more precise, amino acid number 69 and amino acid number 70, resulted in structural changes in the S1 and the S2 component of the spike protein of the coronavirus COVID-19. And you can see this change in yellow color. You see it here? So you can see that there is morphological changes in the spike protein of the coronavirus COVID-19. Okay. So as I mentioned previously, this changes in the structure of the S protein resulted in a problem. What is the problem? The problem that because of this change, which you see it here, in the coronavirus spike protein, you see this changes here, which I am drawing a black circle around it. This changes in the structure of the spike protein of the coronavirus COVID-19. This change in the structure, it prevented the neutralizing antibodies. You see it here, this is neutralizing antibodies, which I'm pointing out with my laser pointer in brown color. You see this change in the morphology of the structure of the spike protein of the coronavirus COVID-19, which you see it here in yellow color. It prevented neutralizing antibodies that has been formed against the coronavirus COVID-19 from binding specifically to the S1, S2 component of the coronavirus COVID-19. So what that means, you see it here, the X here, it means that the neutralizing antibodies cannot bind to the spike protein of the coronavirus COVID-19. Why? Due to the structural changes in the S proteins of the coronavirus COVID-19. So what that means? It means that this mutation made the coronavirus COVID-19 to be able to evade the immune response. So the coronavirus would, as a result of this HV69/70 mutation in the UK variant of the coronavirus COVID-19, resulted in the capability of the coronavirus COVID-19 or the UK variant of coronavirus COVID-19 to evade the immune response. As you see it here. So antibody cannot bind due to the structural changes in the S protein. So subhanallah, structural changes in the spike protein due to this deletion, which resulted in substitution in amino acid number 69 and 70, resulted in structural changes in the spike protein of the coronavirus COVID-19, so that neutralizing antibodies cannot bind to the spike protein of the coronavirus COVID-19. So then this UK variant can evade the immune response. This immune response could be, for example, your vaccination. So for example, you can give someone or vaccinate an individual against coronavirus COVID-19, but because of this mutation, this will reduce the efficacy of the vaccine to give a high percentage of protection against infection with coronavirus COVID-19. Why? Because, as I mentioned previously, this mutation made the coronavirus COVID-19 be able to evade the immune response. So if you get a vaccine, you generate neutralizing antibodies. This neutralizing antibody, they can neutralize the coronavirus COVID-19, but they are less efficient. So the efficiency of the vaccine, like say for example the Pfizer vaccine, could be 90% for the original coronavirus COVID-19. However, because of this mutation, which is mutation HV69/70, then the efficacy of the, for example, Pfizer vaccine will go down from 90%, for example, to 60%. So this mutation allowed the virus to evade the immune response. The third mutation found in the UK variant P117 of the coronavirus COVID-19, we call it mutation P618H. And this mutation is found only in the UK variant. You cannot find it in the South African or Brazilian variants of coronavirus COVID-19. This mutation, as I mentioned, which we call it P618H, results in changes in the S1 and S2 component or components of the spike protein of the coronavirus COVID-19. This mutation in the S1 and the S2 proteins or components of the spike proteins of the coronavirus COVID-19 will result in the increase in the level of the transmembrane proteases. You remember? TMPRSS2. You remember what I have said at the beginning of this lecture? That on the cell, this is the cell membrane, this is the cell membrane, okay? And then in the cell membrane, we have the ACE2 receptor, you remember? And then we said that the spike protein of the coronavirus COVID-19, which consists from the S1, S2, and the receptor binding domain, which you see here in blue color, will bind to the ACE2 receptor. However, we said that the cells will contain the transmembrane protease, or TMPRSS2. This is an enzyme. This is protease enzyme, will help in the cleavage of the spike peptides or proteins in order to facilitate the entry of the coronavirus COVID-19 into the cell. So I see it here. This is the TMPRSS2 protease. Okay, this protease will help in the cleavage of the spike, which is this one. Okay, they help in the cleavage of the spike protein, and this will facilitate the entry of the virus into the human cell. However, in the UK variants, which has mutation P618H, as I mentioned previously, that this mutation P618H will result in mutation or changes in the S1 and S2 component proteins of the spike protein of the coronavirus COVID-19. This mutation somehow increase the level or result in the increase in the level of the transmembrane proteases. As you see here in this diagram, you can see that there is an increase in the level of the transmembrane proteases. You see the arrow, an indication of the increase in the level of the transmembrane protease, as I'm pointing at my laser pointer. So once there is an increase in the transmembrane proteases, so the cleavage, then there is more cleavage, isn't it? Remember we said that the function of this transmembrane protease is to cleave or cause cleaving or cleavage of a particular part of the S1 and S2 spike proteins of the coronavirus COVID-19. Because there is an increase in the transmembrane proteases, there will be more cleavage of the spike proteins of the coronavirus COVID-19. Then there will be more virus to enter the cells, or increase in the virus entry into the cells. So because there is high, or there is increase in the level of the proteases, there is more cleavage of the S1 and S2 proteins. This more cleavage of the S1 and S2 proteins will result in more virus enter into the cells. So more virus enter into the cells, so this will increase or result in the increase in the viral replication inside the cells. So this will result in increase in the viral load inside the cell. So then, as I mentioned previously, more virus, because there is an increase in the viral load, so more virus will be released from the droplet of the infected individual or in the sneezing. So this will result in more transmission of the coronavirus COVID-19 UK variant. So you see here, you see what we have here. This is the transmembrane proteases, okay? And as I mentioned, because of the increase in the level of this transmembrane protease, which you see it here as a blue triangular, more cleavage of the spike protein. This is the spike protein, more cleavage. So then more virus will enter into the cell. More virus will enter into the human cell. More virus will enter into the human cell. So more virus, it means more replication. As I mentioned previously, this will result in the increase in the viral load inside the cell, for example in the lung cells, and then because there's an increase in the viral load, then more virus will be released from the droplet of the infected individual or in the sneezing. Then this will result in the increase, more virus in the air, more transmission of the UK variant of the coronavirus COVID-19 with the P618H mutation. So here you can see how that mutation, subhanallah, resulted in the increase in the transmission of the UK variant of the coronavirus COVID-19. What you see here, this is in red color. Here we have the spike protein of the coronavirus COVID-19. Here we have this is the viral envelope. This is the coronavirus COVID-19 viral envelope. So this is the viral envelope, and this is in red color, the spike protein of the coronavirus COVID-19, and this is the ACE2 receptor, or the angiostatin converting enzyme 2 receptor. So this is the ACE2 receptor, and this is the cell membrane of the cell. This is the cell membrane. Okay, and here you can see the mutation in the spike protein. You see here we have the N5, for example, 01Y, a mutation which I have described previously. So this is the British variant or the UK variant of the coronavirus COVID-19, and you can see here this is the location. You see it here, drawing a black circle around it. This is the location of the N501Y mutation in the spike gene and corresponding protein of the coronavirus British variant. This is an interesting picture, and this picture shows that when the news spread regarding the appearance of the UK variant of the coronavirus COVID-19 and its high rate of spreading rapidly in England, so what will happen is that many countries closed their borders in order to prevent the UK variant from getting into their countries. And this, because of the closing of the borders, you can see that along queues of the trucks at the borders because of the spreading of news regarding the emerging of the British or the UK variant of the coronavirus COVID-19. We have stated previously that the UK variant or the British variant, it has 3 different mutations. The first mutation, we call it N501Y. The second mutation in the UK variants, we call it HV69/70. And the third mutation in the British or the UK variant, we call it P618H mutation. Now, after the completion of the UK variant or the British variant of coronavirus COVID-19, now we are going to move into the second variant of the coronavirus COVID-19, which we call it the South African variants or P1351 variant of the coronavirus COVID-19. The South African variants of coronavirus COVID-19, it has 2 different mutations. The first mutation, which is mutation N501Y, and the second mutation we call it E484K. The first mutation in the South African variant of coronavirus COVID-19, which is mutation N501Y, is similar to the mutation which I have described previously in the UK or the British variant of coronavirus COVID-19. However, the second mutation of the South African variant of coronavirus COVID-19 is new, which we call it E484K mutation. So, what is the result of the E484 mutation in the gene that codes for the spike protein of the coronavirus COVID-19? The E484K mutation in the coronavirus South African variant results in what we call it changes in the receptor binding domain motif. What that mean? Simply, if we look here at this diagram, you remember we said that the spike protein, it has two components. The first one is the S1 component of the spike protein of the coronavirus COVID-19. The second component of the spike protein is the S2, and then we have the one in the blue color, which we call it the receptor binding domain component of the coronavirus COVID-19. This receptor binding domain, which is, you can see it here as well, RBD in blue color. This RBD, it has what we call it receptor binding motif, which you see here in red color. You see the blue color? This is the blue color is the receptor binding domain, and you see the red color in this blue color. This is, we call it receptor binding motif, which is this one, receptor binding motif. This is the, in red color, is what we call it the receptor binding motif. This receptor binding motif is also involved in the binding of the spike protein to the ACA2 receptor on the human cell. So you'll ask me, what is the relation between mutation E484K in the South African variant with the receptor binding motif? With the receptor binding motif, I will summarize to you or simplify to you in this diagram. You see here, this is the E484K mutation in the gene that codes for the spike protein of the coronavirus COVID-19. This mutation in the E484K will result in changes in the receptor binding motif. Result in changes in the receptor binding motif. You can see the changes here. You see it here? You see here? I'm pointing at with my laser pointer. So the blue color is the receptor binding domain. The blue color is the receptor binding domain. You see here that thick red color, which I'm pointing at with my laser pointer. This is the receptor binding motif. But you can see that because of this E484K mutation, that the structure of the receptor binding motif is changed. It become much, for example, it will become thicker. You see it here? So this is, you see here, this is in the non-mutated. You see it's 10. You see the receptor binding motif is, you see it here, it's 10. However, in the E484K mutation, you can see that the receptor binding motif, which is this one, you see it here, it's changed. There's a change in its shape. So there is a changes in the shape of the receptor binding motif, which you see it here in comparison to this one. You see it here. So this is in the original coronavirus COVID-19. However, in the South African, you can see there is a change in the shape of the receptor binding motif of the receptor binding domain of the spike protein of the coronavirus COVID-19. This change in the receptor binding motif of the spike protein of the coronavirus COVID-19 will result in an increase in the interaction between the receptor binding protein and the ACO2 receptor. You see it here? You see here there is this changes in the receptor binding motif resulted in more interaction between the spike protein and the ACO2 receptor. More interaction. So there is intimate and strong binding between receptor binding motif and the ACO2 receptor. So what that means? It means that more virus will enter the cell. You see it here? So this is the cell, this is the human cell, this is the virus, and because this mutation, which is E484K, and which has resulted in changes in the receptor binding motif, more virus will get into the, or more coronavirus COVID-19 South African variant will get into the cell. More virus will get into the cell. More virus will get into the cell. More virus will get into the cell. This will result in what? Many virus inside the cell. So this will increase the viral replication. This will result in the increase in the viral load inside the infected cell. So then more virus will be released from the cell. Okay? And this virus where? Will go from the lung into the droplet, and then this will increase in the transmission of this mutated or the South African variant of coronavirus COVID-19. So okay, so you can see here, just to summarize what I have said. So you have mutation, which is E484K, results in changes in the receptor binding motif. This change in the receptor binding motif resulted in the intimate and strong binding between receptor binding motif and the ACO2 receptor. This result in more virus enter the cell, so increase the virus entry. Increase of the virus entry to the inside of the cells, followed by increase in the viral or coronavirus COVID-19 viral replication, followed by increase in, because of there is increase in the viral replication, so this resulted in increase in the viral load inside the lung. So then more virus will be released in sneezing or in the droplet of infected individual with the South African variant. So this will result in the increase of the transmission of the South African variant of coronavirus COVID-19. So you can see how a mutation resulted in increase in the transmission of the coronavirus COVID-19. So mutation in the spike protein resulted in more transmission of the coronavirus COVID-19 as I have described. If we look at the third variant of coronavirus COVID-19, which is the Brazil variant, which we call it P1. The Brazil variant of coronavirus COVID-19, it has three different mutations. Mutation N501Y, mutation E484K, and the third mutation we call it K417T. Now mutation N501Y and the mutation E484K is similar to the mutation which I have described previously in the South African variant of coronavirus COVID-19. However, the Brazilian variant of coronavirus COVID-19, it has an extra unique mutation which you cannot find it in the South African variant, which we call it mutation K417T. A mutation in K417T of coronavirus COVID-19 in the Brazilian variant results in more intimate interaction between the spike protein of coronavirus COVID-19 and the ACO2 receptors, and will result in more virus enter the cells, and it will result in increase in the transmission of the Brazilian variant of coronavirus COVID-19. What you see here in this diagram, this is the structure of the Brazilian variant spike protein. Okay, and you can see the mutations in this Brazilian variant, or you can see the mutation in the spike-protein of the Brazilian variant of the coronavirus COVID-19. The first mutation, for example, we have N501Y. The second one here we have E484K. Okay, and then the third one, which is this one, which is the K4170, and this is the, drawing a circle, this is the in yellow color, this is the N501Y, this is the one, this is the second one, which is in yellow color, E4, I'm drawing a black circle around it, E484K, and the third one, which is this one, is the K417T. So all of these are mutations which has been detected in the Brazilian variant of coronavirus COVID-19. So Brazilian variant of coronavirus COVID-19, or P1, it has 3 mutations in the genes that codes for the spike protein of the coronavirus COVID-19. We have a mutation N501Y and a mutation A484K, and as I mentioned previously, these 2 mutations is exactly similar to the mutation seen in the South African variant of coronavirus COVID-19. However, the Brazilian variant, it has an extra mutation, we call it K417T mutation, and this mutation resulted in making the spike protein can have an intimate and strong interaction with the ACE2 receptor, which resulted in more entry of the Brazilian variant of coronavirus COVID-19 into the human cell, and ultimately resulted in more transmission, as I have described previously. The question which is needed to be answered is the available vaccine, like for example the Pfizer vaccine or the AstraZeneca vaccine, are they effective against the coronavirus COVID-19 variants, like for example the South African or the British variant? I'm going to answer this question for you from previously or recently published work or studies. A recently published study suggested that individuals who got infected with the South African variant of coronavirus COVID-19, their ability or the ability of their immune system to generate neutralizing antibodies against this South African variant will be reduced by 10-folds. Also, another recently published studies which conducted small trial on the Novavax vaccine, and this study showed that the Novavax vaccine is less effective against the South African variant in comparison to the original COVID-19 and to the British variant of coronavirus COVID-19. So what that means? It means that the Novavax vaccine is less effective against the South African variant of coronavirus COVID-19. Another published studies showed that the antibodies produced by the Pfizer vaccine are less effective against the South African variant. A recently published study showed as well that the Oxford AstraZeneca vaccine provide a minimal protection against mild moderate COVID-19 in young adults infected with the South African variant. So also the Oxford AstraZeneca vaccine are less effective against the South African variant in comparison to the British or the UK variant. However, there is some encouraging data which showed that individual who they got infected in the past with coronavirus COVID-19 or with the original coronavirus COVID-19, and then they got vaccinated, those individual can produce or produced a good immunity against the South African variant of coronavirus COVID-19. From the above, the global health officials in the WHO, they found that coronavirus COVID-19 virus that carries 3 mutation, which is the N501Y mutation and the E484K mutation and the K417T mutations, like for example the South African variant, they considered this variant, like the South African variant, they call them variant concern. Why the global health officials called the South African variant or the coronavirus COVID-19 that carries these 3 mutations as a variant concern? They call them as a variant concern. Why? Because they have the capability to evade the immune response. This capability of evading the immune response, it means that the current vaccines that we are having now, maybe they are not that effective against this variant concern. That's why global health officials are conducting a number of studies in order to develop new vaccines that will provide a good protection against this variant concern, like the South African variant. As an example, the Moderna manufacturer, the Moderna vaccine manufacturer, they announced that their second vaccine or their second dose of their vaccine will be effective against the South African variant, and they are taking in considerations to develop a vaccine that will be effective against the coronavirus that carries these 3 mutation, which is N501Y mutation, E484K mutation, and K417T mutation. Answering the question regarding if the available current vaccine, if they are effective against the coronavirus COVID-19 variant, will bring me to the end of today's lecture. In today's lecture, I have drawn and constructed a simplified diagrams in order to explain to you and to describe to you the different variants of coronavirus COVID-19. If you have any question in today's lecture or any of my previous lecture, please do not hesitate to contact me by email or by WhatsApp. Thank you.